Hi, everyone. So this is our last part of the day today. And so we're going to continue the discussion that we started before the break, really trying to get at, what clinical disease targets are ideal for focused ultrasound plus immunotherapy. What do we know about the role of different modes of FUS clinically? Have we determined any strategies that we can consider for focused ultrasound treatment of tumors? Things like, and we've heard about some of these today, treating the entire tumor versus treating, portion of tumor, treating primary tumor versus metastasis, and then elements of the trial design as we started to discuss. And we want to make sure to end our discussion today by thinking about, as we did in the previous sessions, what are some projects that are going to be critical to moving this field forward? And again, are there some specific projects you can think of that perhaps the foundation could consider funding, or that we just need to be doing? Are there other things we could be doing together in terms of guidelines or standards? And so I wanted to start by giving all the participants the opportunity to ask questions of the speakers. We heard some really great talks. And so again, if you're interested in asking a question, please use the raise your hand function just so that we can make sure that we're not talking over each other. But we'll start with that. So if the speakers want to come back on camera and go ahead, anyone, feel free to ask any questions. Go ahead, Dr. Wang.
Thank you, Jessica. I was out for a couple of minutes and saw the notes about our paper. Actually, I'm very inspired by this workshop, particularly learning how all histotripsy can alter the micro environment to promote the, switch of phenotype or these local, let's say myeloids, right? So I think those will be all very exciting for us because we design CAR T cells which can be guided by focused ultrasound to turn on and activate them. But it would be great to know, for example, there are studies who already assess the transcriptome or genomic profile or this marker environment upon, let's say, half or histotripsy treatment. What kind of cell type has been changing longitudinally, and what kind of genomic or genetic profile has been changing. Those will be very helpful for us to follow and design controllable CAR Ts. It sounds like different kind of tumor types and different kind of a half versus histotripsy, they may trigger different immune response. So those longitudinal tracking or the transcriptome or single cell RNA-seq kind of study would be really helpful for us. I don't know whether they are already some kind of effort to create database to create different tumor types, and upon half or histotripsy treatment, what evolution or this genomic profile is changing, or cell type profile is changing in the marker environment of these different type of tumors.
Does anyone have an answer for that? Maybe some of our previous histotripsy experts want to weigh in?
I apologize. Can I get Dr. Wang to repeat the question, please?
Yes. So basically, whether there are already efforts, people longitudinally monitor the cell types and transcriptome or genomic profile after the half or histotripsy treatment in the marker environment of tumor across different tumor types.
I mean, I can't speak for the clinical side, but that's something we routinely do preclinically in veterinary patients and in different veterinary models beyond just rodents, but in actual veterinary patients. That has had limited success clinically, at least in the veterinary clinic, just because we're still kind of limited to the time points we can take, and you can only sample the pancreas so much, right? Or the liver so much. Liver is a little bit easier than pancreas to do longitudinal type studies like that, but you have to be careful when working with clinical patients. I'm assuming even more so on the actual clinical side.
Thank you. Yeah, I thought those kind of study would be really helpful and perhaps require some team effort, individual lab probably will be difficult.
Yeah, as a clinician, I'm not really aware of anyone who's done a nice longitudinal genomic study after FUS treatment. There is now a clinical tool using circulating tumor DNA. And so some other research areas have looked at changes in mutations in the circulating tumor DNA over time following various different treatments. And you can see lots of evolution, but you can also see quite a lot of heterogeneity emerging over time after various different treatments. So I think it would be easy to include in future focused ultrasound studies, but I'm just not aware of anyone who's done it yet.
The group of Joseph Frank has done studies like that in preclinical models. So there is a wealth of information on the regulation of chemokines genes, cell subtypes invading the tumor longitudinally and in different tumor models. So you can have a look at their papers. There is quite a wealth of information now. The challenge is to amalgamate all this information to understand better what's happening, I think.
Thank you. That would be very helpful. Yeah, I mean, whether the FUS Foundation would help also to collect those information, create some kind of database for people to easily, find out those information if they are available. That would be hugely helpful for the community.
There is an interesting variability in the regulation of the genes, of chemokines, of cytokines, of tumor, of TILs in the tumors. Yeah, it's nice to read all these papers.
Can I respond at least?
Sure.
Yeah, so yes, and in fact, you may wind up with the same outcome and completely different genomic changes occurring in the tumor. And I think that's really something we haven't explored, that if we compare, say, the B16 to the 14-1 tumor lines, we are seeing different proteomic changes. I mean, we are actually seeing a lot of interleukin changes as well as interferon gamma and IP1, IP10 occurring early, but they do change temporally over time, and it also depends on exactly when you do the experiment. I think this is something that's missing here. I mean, if you do the experiment at day 5 and decide to sonicate with 6 megapascals at 1 megahertz, you wind up with basically a mechanical effect, not a ablative effect, and there's really no thermal effect that we're seeing, certainly less than 2 degrees C, and do not see any necrosis, increased necrosis. But in fact, the changes that you see in the tumor in the proteomic and the genomic environment are quite dramatic, depending upon if you do the experiment on day 5, day 7 or 8, or day 10 or 11, and that corresponds to sort of a 5 millimeter diameter tumor to a 8 millimeter diameter tumor to a sort of a 10 or 12 millimeter diameter tumor. And so that's, I think, really important. The other thing that I want to mention is we've been studying recently, and this may complicate what happens clinically. And in the preclinical model, we actually went ahead and ordered animals from 3 different vendors, the same black 6 mice from 3 different vendors, and implanted tumors in their flanks and found different proteomic, genomic, and growth profile responses in response to focused ultrasound, mechanical focused ultrasound. I think that, and we're now doing the DNA analysis to find out whether or not they're really truly all black 6 mice from Charles River, Jacks, and Taconic. And the real questions that's going to come up, is it metabolic or microbiome change causing this effect, which we don't really think is possible, because they tend to stay in the facility for at least 10 days before we do the experiment. But is it something else that's going on here? And this is going to be reflected, is this going to reflect the tumor heterogeneity or response to focused ultrasound in a general population? And I think that's also something that we need to look at in the future.
May I just ask if tumor heterogeneity is the cause of all this, how do you solve something like this?
I mean, the best way to do is large numbers of animals and have it repeated in different places. I think that's the one. I mean, we're spending a lot of time on reproducibility now, and going into larger and larger N's in order to be able to start to get an idea what the what the heterogeneity is. I think that you're going to see this not only with the immunoscores, that if you want to take a look at the histology and where the distribution of cells are in the tumor, but also how they respond to from the changes in the tumor microenvironment. I mean, I'm really surprised. I was actually shocked to see that a same B16 tumor planted on the same days, all at the same size, being sonicated at the same time, essentially wound up with proteomic responses that are completely different, and growth responses that are completely different, where essentially in two of the three vendors, you wound up not getting a response, but only in one, in the Charles River group, did we actually see a response where we saw changes in the tumor microenvironment. I think that's going to be probably translate into what we're going to see clinically, is that there's some people who are going to basically who are going to respond, some not. And I think we also have to look at the definition of response. We're not looking for CRs in any of this sort of situation unless you completely burn away the tumor. You're looking for partial responses or a delayed in growth, so that some other things can be done. This is going to probably serve as an adjuvant approach or neoadjuvant approach as compared to a complete treatment.
Well, I think in this discussion, we're seeing that obviously there's a lot of variabilities that we're noticing in the preclinical studies. We're going to see even more variabilities in the clinical studies. So at this point clinically, how confident, or are we confident at all, that we can say, this type of focused ultrasound is, be it histotripsy, be it ablation, is the optimal type to use for this patient population or this tumor type? What kind of data do we need? Do we need more preclinical data, or are we going to have to get more data clinically? And how do we kind of determine and think about what that is to help us make those decisions? Because there are a lot of, there are some sites that have only access to one type of focused ultrasound, but there are other sites that have access to, various focused ultrasound systems, and would really want to make sure that, they're making their decisions on what type of focused ultrasound to use based on, what we know could be the best clinically. So I just wonder if anyone has any thoughts on what they've seen. Dr. Vidal-Gervais?
I think we need more data, more clinical data, and more basic data. We need to focus on the clinical data that we have going for ablation, and we need the data that we may have on not doing just ablation, just having partial ablation plus normal tissue, and see what happens in the lab and in the basic research. But then we need to do more clinical data on full ablation. And we have, our amount of cases that we've done with histotripsy is very low. It's only 8 cases. And we're now doing a multicentric study. We have at this point, I would say, more than double of these cases studying, but still the data is very premature. I cannot disclose anything. And we're having, some abscopal effects or anything similar, but we need to see how this data matures and how this data appears. But I think that is still very early to go for anything.
Does anyone else want to weigh in? Or in terms of,
Tim here. I think we're facing a situation that we don't even know how much stochasticness there is in any patient's response. We have very premature data coming out of the breast 48 study with RNA-seq that's suggesting we're having to do quite a lot of modeling the RNA sequencing so that we can diminish the influence of each patient's tumor's fingerprint first, and then seeing what is the immune response overlaid on top of that. So I think we're way too early to be saying that we can generalize. I think with the AM003 study, which Lynn talked about, we're going to have an opportunity at a very early time point to biopsy the sarnicated tumor to understand how consistent across patients and across diseases do we get a similar response, or is it very, very different according to the patient and the disease? And so I think once that data starts coming out, then we'll actually see how much of a barrier we're actually facing to answer that question.
You know, in terms of the aspects of trial design that we think should be incorporated into all of our clinical trials, do we have any thoughts on that? I mean, we were just talking about sort of the time points after focused ultrasound when you would be taking biopsies or blood samples and doing immune assessments. Do we have any, or does anyone have any strong opinions on those time points? I think I'd also direct you all before tomorrow to look at a link that Kelsey just put into the chat, which is the guidelines, the immune assessment guidelines that the foundation has put forward. These were developed with our scientific advisory board, and many of you in this workshop participated in that development. And these are guidelines for clinical and preclinical studies, and they're kind of meant to be a minimum set of parameters and kind of metrics to use for immune assessment. But that's something that we're going to talk a lot about tomorrow. And so I'd encourage you all to kind of think about what your thoughts are on what we already have and what perhaps could be changed or incorporated into those guidelines. So if anyone has any thoughts on any of that, go ahead, Ray. Go ahead.
You mentioned design considerations here, and I think we all would like to think that with immunotherapy, that immunotherapy earlier in the course of a solid tumor is probably going to work a little bit better before we have all of the heterogeneity that develops later on, all of the molecular instability, the chromosomal instability, the genome becomes more and more unstable as you get into late lines of therapy. We talked about what multiple lines of chemotherapy and multiple lines of immunotherapies or steroid therapies and GBM, all those things make really late line trial design a little bit harder from an immunologic standpoint. And of course, all the companies developing immunotherapy checkpoint inhibitors have found generally greater activity in earlier lines of therapy with metastatic solid tumors kind of across the board. So of course, we'd love to be doing more of our FUS immunotherapy studies earlier on as well. We ran into regulatory problems with the FDA when trying to propose an ablate and resect in early stage disease study. That's like most things, the FDA wants you to try in sort of the last line and most heavily pretreated, most refractory patients first. And so that regulatory issues factor into how we design these trials and into then how we can follow up and how long patients will be expected to survive after participation in a trial at their late stage. So I think there's a lot of creative ways to work with FDA and regulatory authorities in designing these trials. And so that is a big part of what we've gone through. I think we heard in both of the presentations earlier that window of opportunity is a great, an ideal place where you can treat a tumor and then resect the tumor. That's a great opportunity, but that took quite a lot of negotiation, at least for our window of opportunity trial. And I suspect the other groups have seen the same.
Dr. Gervais, I think you're muted.
Sorry. I think there is a good opportunity, a good clinical opportunity in a setting that we may consider that probably may overcome this problem, as Patrick was saying, is that the patients that are in a waiting list for liver transplant, those patients are normally not being treated or are not being offered anything as a protocol, or maybe some of them may have an ablation, may have a REF, may have anything. those patients may be candidates to do on a study, to do this type of histotripsy, make the studies of immunotherapy, and then when they're going to be transplanted, we can have the liver and see what has happened there. And this probably may offers a special situation, clinical situation, when we can work on that. It's a suggestion.
If I can make a comment, it's just a very hard thing to apply standards for all these tumors, right? For melanoma, it's pretty straightforward to be able to poke and get samples for patients. Whereas with the glioblastoma, surgery is very expensive. To be able to give more than one specimen and one biopsy is probably going to be very difficult. And so, and they're all kind of moving targets. So, I think it's always interesting if you look back and you look at some of the big pharma companies like BMS, right? They're the ones that were, with Merck first in line with all these immunotherapy strategies. And one of the things that I've taken away from them is most of the times they say blood has been disappointing. And we talk about all these great protocols about getting blood and checking the periphery. And glioblastoma, we think it's even harder unless you're doing stuff like CAR T cells like Dr. Okada is. Showed, but it's interesting if you start looking back at the at the output of all these correlative studies, and this industry has had more resources and they put tons of money. Blood seems like it's been the most disappointing, and it seems like going after the tumor is where it's going to be at. And so I take blood from all my clinical trials still, and it's like industry said, I've had pretty disappointing results. It's really only when you get to the tumors. And so, perhaps it is more, as we figure out our resources and we may be trying to figure out ways to get to the tumor, and we may have to require, 2 surgeries for people or something to that extent, or multiple surgeries.
I'll respond to that because I think, everybody's looking for the answer, right? And I think when you really look at what it is, the tumor is so informative. Because it's the disease state. But then when we think about what our tumor samples are, especially in a metastatic setting, it's a core needle. So it's a fraction of a fraction of a fraction. And then when we do high dimensional imaging or other assays, it's a portion of that. So we have learned a lot from that. And the baseline tumor samples can be informative in terms of what the crosstalk between the tumor genetics and the immune features are, at least to know if you have an immune desert. But I actually think there's more and more in blood that's really come out. And it's really, I think the issue isn't, having worked in big pharma and other places. And one of the things that took me to Parker was that people are really organized functionally. And as I started in translational medicine very early, like before we even called it that people were so focused on a technology. So genomics was hot. This was hot. So you had the genomics people or the pathology people or the proteomics people. And it's really the totality of the data. And so, using, and we have such a luxury. I would tell people, I know when I was considered myself to be way cool, when we did 8-color flow, to have access now to these panels that are really 40-plex, where you can characterize immune features and immune system characteristics, and doing longitudinal assessment and then using these broad panels with proteomics. You can get some really interesting data sets. And I think it's the discipline of having people that can collect all the data, integrate the data, and then have the in-depth immunology to know what an antigen experienced T cell is versus an antigen primed dendritic cell. And, you know, all of those markers really takes a village of expertise from both how to gate that and take the proteomics with all the different features to look at dendritic cell markers or T cells, or what a CXCR5 positive CD4 is, and that what's the follow-on effector study. And that's where I think if we really come together as a multidisciplinary team, we may be able to see more there.
No, I think it's a great point. And, you know, I fully acknowledge as technology gets better, we'll probably have better detection for blood. But I also think you brought up a really important point, which is bioinformatics. And it's massive, massive amounts of data. And so as we're doing flow cytoph and flow cytoph version 2 and single cell sequencing, that's going to be even you know, higher, you know, dimensions. I think that's where a lot of us are going to hit some obstacles, because it's, you know, not all of us have, you know, I feel lucky I have some of those capabilities, but not all of us have that. And, you know, it's interesting. I'm encountering bioinformatics people who've never taken a biology course because there's just a shortage, and I have to educate them. So, you know, I think if there's going to be resources, trying to create some sort of core, and maybe the Focused Ultrasound Foundation can think about that, like trying to create some sort of infrastructure for these massive amounts of data and dimensions of data that are going to come through. I think that'll be just as critical as having these protocols that we've designed.
Any other comments there? Oh, go ahead, Dr. Wang.
Thank you, Jessica. I totally agree. You know, want to echo Teresa and Michael. You know, interdisciplinary collaboration and team effort probably will be needed. You know, we first need to know the possibly composition of these microenvironment tumor and the cell types, extracellular matrix, right? And then we want to, you know, dig deeper into the transcriptome, you know, genomic profile. All these, you know, definitely require a lot of, you know, cross-disciplinary, you know, collaboration, like bioinformatics and the immunologist and the cancer biologist. Just want to chime in a little bit.
So I think, you know, as we have about a little less than 10 minutes left today, get you all thinking, and some of this was just maybe alluded to, some of the steps and projects that are needed to move us forward. Are there places where the foundation can be particularly helpful? But sort of thinking about, you know, where we are now clinically, and if we are designing new clinical studies moving forward, are there specific tumor types that we should be thinking about? You know, are we thinking about areas, diseases where we know that immunotherapy already works and focused ultrasound could perhaps, you know, add to that, or help more patients be responders there? Or are we thinking of, you know, diseases where there isn't a good impact of immunotherapy now, and focused ultrasound could perhaps be something that could strengthen that? So that's just sort of one guiding question. But any thoughts on kind of where we should be taking our next steps in terms of clinical projects? This is for anyone to weigh in.
I mean, the one thing I just, not for being forward looking, other than to have the patience to get the data. The trial designs that the speakers gave earlier, and I really have to applaud them. I think that that's a gold mine of data that's in progress, and maybe really trying to make sure you maximize the analyses that we heard. I mean, you have a treasure trove of different approaches with combinations or not, and different solid tumors that you can really learn something with different clinical hypotheses, and to that is the one thing, is that to do all the analysis does take time and dedication. And we often, we want to publish the results of our papers fast in clinical studies. And in these cases, sometimes the clinical endpoints will read out a lot faster than you can do all the biomarker work.
May I have one comment? This is Hideo Okada from UCSF. So for the brain cancer perspective, I would like to have a more interaction with people who are working in the space of not necessarily in cancer, but disease entities like Alzheimer's disease or CNS autoimmunity using focused ultrasound. In general, for us to study and promote immunotherapy for brain cancers, I personally, or many people also, have been taking lots of insights from the lessons and the discoveries from the area of autoimmunity in CNS, such as multiple sclerosis or experimental allergic encephalitis, EAE models. So knowing that FUS technology is being applied for disease entities such as Alzheimer's disease, I would love to have more interaction with people who are working in that space so that we can better understand more effectively about what FUS can do on CNS environment.
Definitely. Anyone else?
Hi.
Oh, yes, go ahead.
I just wanted to also reiterate some of the points brought up about the wealth of data that's coming out from the trials, and just specifically within the context of pancreatic cancer. It can feel a bit lonely doing a trial, particularly an intervention trial where you're gathering data and biological material, but there aren't many other pancreatic intervention trials going on across the world, really. And so having something like the foundation as a platform for sharing our experience, in particular the challenges with other trialists, and probably more importantly, we all have our strengths, particularly when it comes to measuring biological endpoint data. And it might be that other centers, say with pancreatic cancer, may be stronger in certain areas. So particularly with the blood parameters or indeed histological endpoints. And so collaborating and sharing, I think, would be a really useful way forward, particularly in the challenging biological sample tumor types like pancreatic.
That's great. And that's part of the goal of these sorts of meetings as well, is to try to get us sharing, but we can definitely come up with other forums for people to share their challenges and discuss things like that. Anyone else? Or we can end a few minutes early. All right. Well, today was a fantastic first day. Thank you all for the great discussion. We'll be coming back tomorrow morning, 10 a.m. Eastern time, for more great discussion. We'll be talking about a lot more about the metrics of success, and particularly how do we do immune assessment in preclinical and clinical studies. We'll be talking more about kind of using the aspects of focused ultrasound that enhance delivery of drugs, and how that could be used specifically in tumors like glioblastomas and pancreatic cancers, as we were kind of talking about earlier. So we'll hear some more about that, and then we'll have an opportunity as well at the end to kind of revisit some of these roadmap items, the kind of next step projects that we've come up with, and talk more about that, as well as some of the other challenges and and how the foundation and our partners can try to help address those challenges. So we'll look forward to seeing you all, and have a great afternoon and evening.